Hello everyone. Today we'll be solving Cambridge International AS and A level Biology Paper 2 AS level structured question, May June 2021, variant 2-1. Question number 1. Figure 1.1 is a transmission electron micrograph of cells from duckweed Spirodella oligorhiza. Okay. Calculate the actual width of the cell labeled X. Write down the formula you will use to make your calculation. Show your working and give your answer in micrometers to 1 decimal place. So the formula, here we have to calculate actual width. So actual width is equals to image width divided by magnification. Now, cell labeled X, this line labeled X has a value of 1.9 centimeter. So A is equals to I divided by M. Here I is 1.9 centimeters, so we're going to multiply it by 10. And then we're going to multiply it with 1,000 to make it into micrometer. And the magnification is 4,275. So the answer is going to be 4.4. So 4.4 micrometer is the answer. B1. Table 1.1 lists some biological molecules found in plant cells. Complete table 1.1 by choosing one letter from figure 1.1 that indicates a cell structure where each biological molecule is found. So DNA, biological molecule DNA is found. Well, obviously we'll find DNA in the nucleus. So B will be DNA. The next molecule is cellulose. Cellulose will be found in the cell wall. And we can see E represents cell wall. Phospholipid. Phospholipid will be found in membranes. And we can see A represent nuclear membrane. Nuclear envelope. So phospholipid A is the correct answer. Histone proteins. Histone proteins can also be found in nucleolus. So B is nucleolus. And we can find histone protein in nucleolus as well. So B is the answer. Next question. State the name of the cell structure visible in figure 1.1 where ATP is synthesized. So in figure 1.1, where ATP is synthesized, we can see chloroplast. So chloroplast. Name a cell structure that produces mRNA. Cell structure that produces mRNA will be nucleus. So nucleus. Describe the evidence from figure 1.1 that show that the image is a transmission electron micrograph. So in this transmission electron micrograph, all right, how can we tell that this is a transmission electron micrograph? Is that we can see the section with a very high resolution. As you can see, we can also see the stacks of grana in this. We can see the nuclear envelope clearly. All right, grana is only seen in transmission electron micrograph. So that is an evidence. And high magnification, all right, higher than the light microscope can be seen, which is 4,275. We know that the highest useful magnification for a light microscope is only 1500x. So it is more than that, and it is still very clear and visible. So thereby, this is, from a transmission electron micrograph. Question number 2. Figure 2.1 shows 3 molecules of water. We can see the water molecule and we can see hydrogen bonding between the water molecule. The question says, describe the hydrogen bonding that occurs between the water molecules shown in figure 2.1. You see, hydrogen bond is a weak bond. Each oxygen forms 2 hydrogen bond and each hydrogen forms 1 hydrogen bond. The attraction between oxygen atom of one water molecule and hydrogen atom of another forms a hydrogen bond. Water is dipolar, as electrons are unequally shared between oxygen and hydrogen. Part B. The human enzyme salivary amylase is composed of one polypeptide. Example, figure 2.2 represents the structure of a molecule of salivary amylase. All right. We can see alpha helices, and we can also see beta pleated sheet present. Explain the role of hydrogen bonding in maintaining the secondary structure of proteins such as salivary amylase. We know hydrogen bonds maintains alpha helices and beta pleated sheet. Question B, part 2. Explain the role of hydrogen bonding in maintaining the tertiary structure of protein such as salivary amylase. Hydrogen bond forms between the R groups with amine and carboxyl groups. Hydrogen bond helps to stabilize further folding of amylase protein. Hydrogen bond may form in amino acids far apart in primary structure. Hydrogen bonds helps to maintain globular shape and active site. Question C, outline the importance of water as a solvent in plants. Water dissolves mineral salts and ions and sugars. Mineral ions such as nitrate, phosphate, and potassiums are carried dissolved in xylem. Metabolic reactions occur in water. Water dissolves carbon dioxide and oxygen needed for respiration and photosynthesis. Question number 3. Weisking tubing can be used to investigate the properties of cell membranes. A student carried out an experiment to investigate osmosis using Weisking tubing. An outline of the investigation is shown in figure 3.1. Measuring rule, Weisking tubing, meniscus, sucrose solution, top of the knot in Weisking tubing, 15 cm cube of sucrose solution, 0.9 mole per dm cube, which is placed outside. All right. 6 pieces of Weisking tubing were filled, 10 cm cube of different concentrations of sucrose solution, ranging from 0.0 to 2.0 mole per dm cube. The height of the meniscus of each solution in the Weisking tubing was measured. The pieces of the Weisking tubing were put into test tube containing 15 cm cube of 0.9 mole per dm cube sucrose solution. After 20 minutes, the pieces of Weisking tubing were removed from the test tubes and the height of the meniscus in each was measured. Okay, so we can see the concentration of sucrose solution inside Weisking tubing. When it is 0.0 mole per dm cube, the difference in height of meniscus after 20 minutes is -12. So we can see water moves out. In all of these 3, water moves out. And from 1.2 to 2.0, we can see in all of this, water moves in. Okay. Now, part A. The Weisking tubing used by the student was not permeable to sucrose. Explain the results shown in table 3.1. So we can see the concentration of sucrose from 0 to 0.4 and 0.8 mole per dm cube. Water moves out of the Weisking tubing. concentration of sucrose 1.2, 1.6, and 2.0, water moves into Weiss King tubing. There is a net movement of water in all Weiss King tubing we can see in this experiment. Water enters Weiss King tubing. The external solution has higher water potential than the contents of the Weiss King tubing. Water moves down when the water moves down the water potential gradient. Part B. When red blood cells are placed in water, they are destroyed by bursting. The student also investigated how red blood cells are affected by immersion in solutions of sodium chloride of different concentration. Blood samples of the same volume were added to the solutions of sodium chloride in separate test tube. After 10 minutes, the student took 0.1 cm³ of the blood samples from the test tubes and estimated that the percentage red blood cells that had burst in each blood sample. Figure 3.2 shows the student's result. Percentage of red blood cells destroyed by bursting, and concentration of sodium chloride solution. As we can see, from 0.0 to 0.04 to 0.04% concentration, all the red blood cells were burst. After that, as the concentration increased from 0.04 up until 0.14, all right, as the concentration increased for sodium chloride, the percentage red blood cell that were bursting, they decreased to 0 at 0.14 sodium chloride concentration. Any further increase in concentration did not cause the red blood cell to burst. So we can assume that the red blood cells were crenated because they lost water. The question says, describe and explain the effect on red blood cells of immersion in different concentrations of sodium chloride as shown in figure 3.2. So basically, at concentration less than 0.04, at concentration less than 0.04 mole per dm cube, all cells were burst, all cell burst. In concentration less than 0.04 mole per dm cube of sodium chloride, water moves into the cell down the water potential gradient. The cell increases in volume and the red blood cells burst because they do not have cell wall. At concentration 0.04 to 0.14 mole per dm cube, decreasing percentage of cells burst between 0.04 and 0.14 mole per dm cube, water potential gradient into the cell decreases. At concentration above 0.14 mole per dm cube, no cells burst. At high concentration above 0.14 mole per dm cube, water leaves the cells. The cells show crenation. Question number 4. The induced feed hypothesis and the lock and key hypothesis are used to describe the mode of actions of enzyme. Explain the induced feed hypothesis. So basically, an active site of an enzyme is not fully complementary to its substrate. So the active site changes shape slightly to fit around the substrate and forms enzyme substrate complex. The active site then returns to original shape on release of the product. Change of the shape to give complementary fit also lowers the activation energy of the reaction. Part B. Radish plants contain the enzyme peroxidase that catalyzes the breakdown of hydrogen peroxide. Students investigated the effect of increasing the concentration of hydrogen peroxide on the activity of peroxidase extracted from radish. The results of their investigation are shown in figure 4.1. Initial rate of the reaction in micromole per minute, and concentration of hydrogen peroxide in millimole per dm cube. We can see that the initial rate of the reaction, the maximum initial rate of the reaction that we have reached is 5.6 micromole per minute, and this occurs at a concentration of 0.35 millimole per dm cube hydrogen peroxide. So up until that, hydrogen peroxide was the limiting factor. Explain the effect of increasing the concentration of hydrogen peroxide on the initial rate of reaction as shown in figure 4.1. The rate of the reaction increases as substrate concentration increases to 0.35 millimole per dm cube, as some active sites are not occupied. Higher the concentration, the more collision between enzyme and active site, and above 0.35 mole per dm cube, all active sites are occupied. At low substrate concentration, enzyme concentration was not limiting factor. However, at higher substrate concentration, enzyme concentration becomes the limiting factor. Part 2. The students determined the Km for reddish peroxidase as 0.1 millimole per dm cube. With reference to figure 4.1, describe how they determined the Km. So Km, all right, in order to determine Km, all right, first of all, we need to know the Vmax. We can see the Vmax is occurring at 5.6 micromole per minute. All right, the V maximum velocity that the enzyme is working. And the Km is the substrate concentration, which is exactly half of that. So basically 5.6, half of 5.6 will be 2.8. So this will be 2.8. And as you can see at 2.8, it is 0.1 millimole per dm cube of hydrogen peroxide. So this is the Km value. Part C, a further investigation found that the Km for carbonic anhydrase enzyme is 12 millimole per dm cube. Describe the role of carbonic anhydrase in the transport of carbon dioxide. So basically, carbonic anhydrase enzyme catalyzes the reaction between water and carbon dioxide in red blood cell to form carbonic acid, which then dissociates to form hydrogen carbonate and hydrogen ion. Hydrogen carbonate ion moves through the membrane protein from the red blood cells into the blood plasma. The activity of the enzyme carbonic anhydrase maintains steep concentration gradient for diffusion of carbon dioxide into red blood cells. In lungs, carbonic anhydrase converts the hydrogen carbonate back into carbon dioxide so that carbon dioxide can be excreted. Question number 5. Much success was made in reducing the number of cases of malaria between 2000 and 2015. Explain how malaria is transmitted. Okay, female Anopheles mosquito act as vector for the malarial parasite. Female Anopheles mosquito takes blood from an infected person, and that's how they start carrying the malaria parasite. And then when it takes blood from an uninfected person, it passes the plasmodium in saliva, which it uses as an anticoagulant. Part B. Diagnostic test strips for malaria contain monoclonal antibodies. The test strip detect antigens produced by the pathogens that cause malaria. Figure 5.1 shows stages in the production of monoclonal antibodies. The information in 3 of these stages is incomplete. Complete figure 5.1. Malarial antigens are injected into mouse. Immune response occurs in mouse and specific dash divide to form many plasma cells. Obviously, B lymphocyte divides, so it will be B lymphocyte. Plasma cells extracted from spleen of mouse and myeloma cells, plasma cells dash with myeloma cells. So they are basically, fused together, obviously, fused together, all right, with myeloma cells. And this forms hybridoma cells. C, part C. Figure 5.2 shows the diagnostic test strip for malaria. We can see C, and T. So T is the basically test. Okay, a sample of blood from a person suspected of having malaria is put into the well labeled S. A buffer solution is put into the well labeled A. The buffer solution moves the blood towards the results window. The line at position C indicates that the test is working correctly. A line at position T indicates a positive test result for malaria. State 3 advantages of using test strip for malaria, such as those shown in figure 5.2. So basically, the results are accurate and it relies on monoclonal antibodies. The diagnosis is very quick. It is a relatively low cost solution. It is easy to use and does not need a highly trained professional. It does not need specialized equipment, reduces unnecessary use of anti-malarial drugs on patients, those that don't have malaria. Guys, even though a lot of options are given, only 3 points, any 3 points from these particular points will suffice for the answer. Part D, the highest number of cases of malaria in sub-Saharan Africa and Southeast Asia occurs in Southeast Asia. Discuss the factors that determine the global distribution of malaria. So basically, the habitat of Anopheles mosquito is tropical, warm, and humid climate. Anopheles mosquito only lives in humid condition. Warm temperature is needed for the development of the mosquito larvae. Mosquitoes require bodies of still water for breeding and places where there is sufficient rainfall. Obviously, the rainfall, provides the still water. All right. Anopheles mosquito are able to spread where they become resistant to insecticide as well. Anopheles mosquito can spread where control measures are not implemented fully. Malaria can spread if infected people migrate from areas with high rates of malaria. Question number 6. Lysosomes are cell structure that contain enzyme known as acid hydrolases. Figure 6.1 shows some processes that occur in animal cell. So we can see bacterium. Bacterium ingested and formed vesicles. All right, the vesicles then are fused with lysozymes. All right, and lysosome digests it. Okay, the lysosomes are also attaching to old organelles such as mitochondria. The lysosome is digesting the old organelle, and digested product is left behind. We can see in part A rough endoplasmic reticulum, and we can see Golgi apparatus in part E, and we can see vesicles formation from rough endoplasmic reticulum. Okay. Name the cell structure labeled A and E. So basically, A is rough endoplasmic reticulum. And E is Golgi body. State the function of the structured label F. The function of structured label F. So F are vesicles. They transport enzymes from RER to Golgi body. Part C, name the process by which bacteria are taken into the cell at C. Phagocytosis. With Part D, with reference to the process occurring at B and at D, figure 6.1, outline the role of acid hydrolase in lysosomes. So basically, acid hydrolases break down the bacteria and or out defective organelles. Acid hydrolase speeds up hydrolysis of proteins to amino acid so that amino acid can be reused within the cell. Part E. Carrier proteins in the membranes of lysosomes maintain a lower pH than the surrounding cytoplasm by moving hydrogen ions. Suggest how the carrier proteins maintain the lower pH within the lysosomes. So the carrier proteins, they can pump hydrogen ion into the lysosome against the concentration gradient. This is an active transport and uses energy from respiration. Thus, they can keep the pH very low. Okay, guys, that's all for today. Inshallah, see you in the next class.